The basic local alignment search tool, or hosted by NCBI, is it basically finds sequence similarities between uploaded sequences. It will essentially give you a percent identity that can help you identify uh, a particular species that your DNA or protein of interest could be similar with. It can be extremely useful in daily life if you work in a biology lab where you would need to uh, where you may come across clone sequences or uncommon sequences, and to be able to identify their evolutionary makeup. Blast can be essentially there are several types of blast, such as the nucleotide blast, which will match everything against DNA or RNA. Protein blast, which is essentially matching protein sequences, and the more uncommon ones, which is the blast X, which works for translated nucleotide sequences against proteins, and the T blast N, which is protein sequences searched against the translated nucleotide sequences, opposite to the one I talked about before. So essentially, there are multiple ways you could put up a sequence in BLAST. One being, if you have a FASTA file of your sequence, you could just upload it onto NCBI BLAST, and you can have a number of parameters that it can search against. So if you knew that your sequence belonged to human or mouse, you could definitely tell the program to just blast it against that particular sequence. Coincidentally, you can also figure out how variant or how divergent or similar your sequences are against other genomes, which could give you a better understanding of whether that particular region of DNA or that particular protein sequence is more or less evolutionarily conserved. It also gives you specific nucleotide differences, which can help highlight what parts of that particular sequence are less conserved evolutionarily than the remaining parts of the protein. Thank you.